Welcome to another episode of Medicine 3.0. This week, I'm very excited. I've got Christin Glorioso. She is a physician. She is a PhD. She has a company called NeuroAge, and what they do is they have several tests to help you understand what is your risk of dementia and Alzheimer's, and really lay out a plan for you to be able to optimize your brain health. So we go deep. We talk about the science behind this, her story, her family history, and why she's really passionate about this. I think you're really going to enjoy it. Christin, welcome to the podcast.
Thanks, Jared. I'm happy to be here.
Awesome. Well, I'm excited to talk to you. I think that, brain health is one of those things that people really, really care about. But let's start out by getting to know you a little bit. Tell us how you got where you're at, knowing what you know.
Yeah. So I'm a physician and a neuroscientist by training. And the reason I started the company is really my own family history of Alzheimer's disease. So when I was a kid, my grandmother suffered with Alzheimer's. I have other family members that are suffering as well. And I found out recently that I have a genetic risk factor for Alzheimer's disease myself. So I have an ApoE4 allele, which puts me at higher risk. And so my life has been dedicated to coming up with better solutions for people who have high risk of Alzheimer's, which if anyone lives long enough, that's really all of us, because there's as we get better at treating heart disease and diabetes, more and more people are going to be suffering with dementia as we live longer. So it's a huge problem. And I have been studying what happens during normal brain aging that puts us at higher risk for the last 20 years of my career. So I was an MD PhD student at University of Pittsburgh and Carnegie Mellon. And then I continued to study brain aging at MIT, where I did my postdoc and stayed on as a scientist before starting NeuroAge about 3 years ago. And the company is aimed at helping people prevent dementia in the future by slowing down their brain aging. So there's this huge actionable piece that we have control over. It's about 40% lifestyle, our risk of dementia in the future.
Very cool. I'd love to dig in and hear a little bit more. Did you always know that you wanted to be a doctor? Did you always know that you wanted to be a scientist? Obviously, it sounds like your grandmother had like a pretty big impact. What can you tell us about that?
Yeah. So I made that decision, I think, in college. So when I was a kid, I actually loved creative writing and art. I also, love science. So I come from a science background. So my father's also a scientist. So I grew up with it in the household and was going into the lab. I was a high school student. So I kind of grew up with science around me, but I didn't really make the decision to pursue science until college. And I had some really impactful classes that I took at University of Michigan. I took a neuroscience class that just really blew me away and solidified my decision to study medicine and to pursue a PhD in neuroscience.
Yeah, and so many people probably don't know what an MD PhD kind of entails. So kind of tell the audience what that kind of path is like.
Yeah. So if your goal in life is to create better therapies or better diagnostics for diseases, like mine was to come up with better solutions for people with Alzheimer's, people will tell you, well, you should do an MD PhD program because it's aimed at creating new medicines for diseases. So took me 9 years. I think that's pretty typical. It's 2 graduate degrees. So you do your first 2 years of medical school, and then in the middle, you do a PhD, and then you go back and do the last 2 years of medical school. And the goal of that program is to really give people the clinical insights as well as the scientific knowledge to bring new therapies and diagnostics to people. And I took a little bit of an unusual path because normally people who do MD PhD programs pursue a residency, and I decided not to do that. I instead took my project with me to MIT and pursued science solely. And the reason that I did that is because there wasn't a whole lot we could do clinically for people with Alzheimer's. So there was a real need for better medicines, and the most straightforward way to do that was to continue working on the research I was working on that ultimately became NeuroAge.
Yeah, I love that. And I love the realization that, like you said, clinically, your passion was Alzheimer's, dementia. There wasn't much clinically that you could do. And so really trying to help invent new, better tools was key. And I think, many people know that doctors study science, but doing the full MD PhD, I actually applied MD PhD when I first applied med school. But it's such a long path. I decided that if I was going to do research, I was going to do a fellowship. And
Yep.
so, yeah, always take my hat off to all the MD PhDs out there that take the sacrifice of, like you said, interrupting your medical training for 4 to 5 years while you do the PhD work, your dissertation and everything, and then back to med school. So quite the path to get where you're at, and really cool. I'm curious, you mentioned your grandmother, you mentioned other family members. When you fell in love with neuroscience, was that always there? Was that, desire to kind of help those suffering from dementia and Alzheimer's, was that always kind of part of it, or did that come later as you kind of moved down this path?
I think that was always part of it. I mean, I think at the core of what a lot of people do is their own family history or their own personal life experience that kind of drives what motivates them. So for me, that was definitely Alzheimer's disease. But I think equally for me as well was just an interest in the brain. I think the brain is fascinating. And, what is mood? What is emotion? What is cognition? What underlies that? So there's also a part of it that I think is pure intellectual curiosity for me.
Yeah, the brain is just fascinating, and I think with artificial intelligence kind of showing us how machines maybe think, and the questions that we start to have about, like, how are we different? I think it really is fascinating. Tell us a little bit about the research that you did at MIT that has kind of been the basis for NeuroAge.
Yeah, so I've been studying human brain aging for my entire career. I actually started that at University of Pittsburgh School of Medicine during my PhD. So I've been working on the same project for 20 years. And Pittsburgh has one of the oldest human brain banks. So when I started graduate school, I thought, okay, we don't really have great medications for Alzheimer's. And in neuro in general, a lot of the medications have been discovered by accident, happenstance. Lithium, for example, comes from or bipolar disorder, comes from people in lithium baths. And they started having less mood symptoms after being in these lithium springs. And so that's how that was discovered. So there's a lot of this that was just happenstance for the medications that we have, and they don't work that well. And a lot of them have severe side effects and off target effects. So, I really thought we need to take an unbiased approach, and it needs to be in human beings so that, we don't have some of the complications from the animal literature. So I was very set on working in human brain. And Pittsburgh has one of the oldest brain banks in their psychiatry department. It's really nicely kept, huge brain bank. And so I joined a lab where I was looking at what happens to the brain during aging that then causes Alzheimer's to occur. So brain aging is the biggest risk factor for Alzheimer's disease. And so we were looking at changes that happen in the brain on a cellular level. So proteins and transcripts that go up and down with age, and whether we can predict how fast or slow someone's brain is aging from those experiments. And so we were looking for genes and pathways that are master regulators of brain aging.
Now,
Yeah.
just to help people understand, when you say brain bank, I'm assuming these are like frozen tissue samples of brains over a long period of time. And then did you also have blood that went along with those samples?
Yes. And it depends on, which set of samples you're talking about. So there's kind of a variety of mix and match, but some of the brain banks have both blood and brain. And so quite literally people die. A physician will go to the scene of an accident or a hospital bed where that person dies and get consent from the family to preserve their organs for scientific research purposes. And then, we get those organs and freeze them, and are able to understand what's happening for people and create better medicines from it.
Yeah. Always such a wonderful gift that people can give after they pass on, to allow us to continue to make scientific discoveries. When you started looking at kind of brain tissue and the blood work of these people, what were the things that you guys were able to kind of elucidate What were you able to understand by looking at these tissue samples?
Yeah, so we made a couple of key observations. So actually, the story of this goes back to my thesis advisor, my PhD thesis advisor, who was Etienne Seville. He's now a professor at University of Toronto, but I was his first graduate student, so he was straight off a postdoc at Columbia University. And he had been actually came from the world of psychiatry. So he was looking at depressed brains versus control brains. And he was looking to say, oh, can we actually see something on a molecular level that makes depression different than someone that does not have depression? And they didn't find anything. So they found no differences between these brains. And so he asked the question, okay, can we see any differences? Because it's a small n,
Yeah.
are we able to see any differences at all? And so he looked at age and sex and was able to see these giant changes with age that happen in the brain. So there's this huge signal. And when we looked at what those changes in levels of transcripts were, and so I'll just explain briefly what transcripts are. So in cells, you have DNA and you have proteins. So proteins make up the working components of a cell, and DNA is the blueprint for making those proteins. And then in between is RNA, so they're the transcript levels. And so that's an intermediary. And so it's a proxy for the amount of protein. So you can look at how much RNA people have of certain genes. And what we saw is that overwhelmingly the RNA levels that go up or down are related to diseases, and they change in directions that you would predict to cause disease. And so it's spurned what we call the age by disease hypothesis, which is as you age, you are losing levels or gaining levels of disease genes that then push you towards disease. So it's actually this gating by the levels of these genes that's causing people to have onset of Parkinson's or Alzheimer's. So maybe to explain an example. So people with familial Parkinson's disease, some of them have a mutation in the Parkin gene that causes Alzheimer's. But Parkinson's. But why does that only occur when you're 63 and not when you're 23? And so we think it's because the other copy, the good copy of Parkin, is decreasing with age. And so it hits a certain threshold that then unmasks the bad copy of Parkin. So that's an example by which changing levels with age are then allowing disease to progress. And so we can track all of those changes. And people with slower brain aging have that happen at a less rapid rate. And so you would expect them to get Parkinson's later or not at all. And so we can kind of tell how people are doing with it.
Yeah. And so that's really interesting too. So what you've been able to find is that, and obviously you don't need brain biopsies to do this, you're able to look at kind of RNA levels that will tell us about protein levels in the brain. You're able to look at those RNA levels in blood and be able to predict how many copies of that protein is there in the brain, either moving you towards dementia or keeping you from moving towards dementia. And how many different proteins through your research, when you look at kind of brain aging, how many different of these transcripts and proteins are you looking at?
Yes, it's about 10% of the genome. So about 2,000 that move like this. So it's a large portion of them. And yep. And it's analogous to other things that I think we're all more familiar with, right? Like increasing cholesterol levels with age. We know that cholesterol itself isn't bad. We all need it. But at a certain point, you have too much of it. And so we're just looking at that for these transcripts in the brain that we can see from blood.
Right. And I like that example of cholesterol. We know that as we age, there are different things that happen in our bodies, that there's increased inflammation, that we don't handle glucose as well. There's all these different kind of changes that are happening. And this sounds like for your brain, these are some of the changes that you naturally see happen just with age itself. But then you can also see kind of people with a higher likelihood start having these things happen right away or at an earlier age. But I think the really interesting thing is that you called out early on in our conversation is that there's like 40% that you can impact with lifestyle. So 60% maybe that's assigned to you at birth with your genetics, and 40% that you can kind of impact. Tell us a little bit more about that.
Yeah, so we found out that we could tell how old people's brains were biologically using levels of these 2,000 RNA transcripts. But we really wanted to know, okay, so what? Does that,
Mhm.
does this way of measuring it actually tie to disease or tie to things people care about like function, memory? And so we followed a group of older adults for 15 years. And then at the end of that study, they agreed to donate their brains to science, and we got those brains. And what we showed is that people that were 5 years younger, so this is then their chronological age biologically. So this is one standard deviation from the mean. So this is totally obtainable. It's the equivalent of getting a B. You don't need an A plus in brain aging. You just need a B, be a little bit above the average. Those people were protected from Alzheimer's. And that was even if they had one or two copies of the APOE4 gene. So they're able to mitigate their genetic risk entirely by being 5 years younger. And even in people without any disease, they had better memory. Their memory was getting worse at a slower rate. They had better mood. They had less pre-Parkinson's signs. So this way of measuring brain aging was really tied to what we care about, which is risk of disease and function.
In that study with those people that you followed for 15 years, what were some of the lifestyle factors that seemed to correlate to younger brain age?
Yeah, so we don't have that data from that particular study, but we do know that from other studies.
Yeah.
And the big six are sleep, exercise, social connectedness, staying mentally active, diet, and lowering stress. So those are the biggest ways that people can prevent brain aging. And a lot of that is taking care of your body, because there's this huge connection between how healthy your body is and how healthy your brain is, particularly in the cardiovascular system. So a lot of what causes heart problems also causes brain problems.
Yeah. So if the easy way to think about it is what's good for your body is good for your heart, what's good for your heart is good for your brain, and you kind of named the 6 things that are good for everything, right? What do you think, like if you have somebody that say they have ApoE3/4, and so not the worst, but still an increased risk for Alzheimer's at an earlier age, and they were to, say they're 45 and they get a test and their NeuroAge says they're 45. And
Yeah.
so they have continued kind of higher risk. What are the kinds of things that you're going to tell that person to focus on to try to make it so the next time they get a test, they're moving towards that B?
Yeah, so we have the general recommendations, and there's more specifics within each of those buckets. You want to get at least 30 minutes of exercise a day. And it turns out that sprinting or getting your heart rate into zone 3 or higher is really good for the brain in particular. And that's because that is the heart rate that releases what I like to think of as plant food for the brain. It's a molecule called BDNF. And when you get your heart rate up, that is when BDNF is released. So, in particular, whatever people can do for that. Then for diet, there's Mediterranean diet has a lot of good evidence for it. Coffee and tea, high vegetable and fruit diet, and then polyphenols, which are found in berries in particular, have a lot of evidence. So there's some specifics around diet, community, want to spend at least an hour a day in social time with friends or family. Lowering stress is a huge one that's hard to do. People can do better at time management or at, optimizing their relationships or their work stress or whatever it is that's stressing them out. So that's another big one, because cortisol is really bad for the brain. It shrinks your neurons as well. So, and then staying mentally active, I think, is the unique one to the brain. So whether some people get that from their job, if they're doing something mentally challenging, if people are retired or don't have a job that's as mentally challenging, getting at least an hour a day in of something that you find hard is important. So whether that's puzzles or hobbies or reading a book, whatever that is. And so those are some of the things. And then we also offer personalized recommendations based on people's biomarkers and genetics. And that's a great part of the NeuroAge test, that we're able to gather data on what's happening with people's RNA levels of genes as well as their genetics and MRI results.
When you say, and I want to dig in a little bit more on all the different tests that you guys do, but let's just talk about like the 5 years younger piece of the data. Is that just based on the transcript test?
Yeah, so that's based on the transcriptomics. But
Hmm.
the cognitive testing also has a long clinical history behind it, showing that people that do better on that testing are less 10-year risk for onset of disease. So we do a battery of cognitive assessments that we call NeuroGames. So there's also evidence for that. And then separately for the MRI piece, there's a lot of emerging and well documented evidence that volume of particular brain areas is predictive of whether or not you'll have disease in the future.
Yeah. It's interesting. I always like to think it would be amazing if, like, we always knew our risk of disease and it was as far out as possible. So we could say, our 30, our 40, our 50-year risk for heart disease, dementia, cancer, all these things. If we could have that, it'd be great. But we don't. We have what we have. And you just named kind of two of those things that I think is really interesting. You've got risk from the transcriptomics test that you're able to run. You have risk from, brain games or neurologic testing. You've got risk from brain volume. And you kind of combine all these things along with genetics, right? So tell us exactly, you've got, is it 5 different things that you run?
Yeah, so there's three different tests, and then genetics is a separate score. So, and then within those three tests, there are some subcategories. So it's NeuroGames, which is the cognitive testing, and within NeuroGames, there's 5 different tests that contribute to that score. The brain MRI, which combines volumes of 26 different brain regions, and then the blood biomarkers test, which is 52 RNA levels of genes. And then we combine that all together to give you a single score using AI and machine learning algorithms.
If someone were to do, say, someone didn't have the means to pay for all the different tests, if they were to do one of those, which one would you tell them is going to be like most predictive and most helpful clinically to tell them how hard they need to be working at changing their lifestyle to improve their brain health?
Yeah, so if you just look at accuracy, the brain MRI is the most accurate, followed by the blood biomarkers, followed by the cognitive testing. But that's also the order of expense. So
Yeah.
I think if you just want quick and dirty ballpark of whether you losing your keys as much as you do or forgetting the names that you're forgetting, if that's normal for your age or something to be worried about, the cognitive testing will answer that question. It takes 30 minutes and is, relatively inexpensive. So, but if you want a really accurate piece and to just choose one, I would choose the brain MRI.
Yeah. It's really interesting as I've kind of dug in on neurologic testing. I've been looking for, what is the cheapest, easiest way to do neurologic testing that is the most predictive. And it's kind of like you said. Unfortunately, the one that's going to give you the best readout for the furthest out is probably the most expensive one. That's just the way the science works sometimes. Sometimes we don't find, the quick, easy, cheap thing that is going to help us. I think one thing that's interesting though is something that you called out again, is that 40%, like, you get to actually have a big impact on your own brain health. And,
Yep.
I'm an ER doctor, so I always just assume the worst thing is happening with somebody. If somebody comes in with chest pain to the ER, I assume they're having a heart attack or an aortic dissection or a blood clot in their lung. Something terrible is happening inside their chest, until I prove that it's not that thing. And it really seems like what people could do is they can live a lifestyle as if they have high risk for dementia until they can afford to actually prove otherwise with the right testing. And then maybe they can eat a little bit more ice cream or but the funny thing is, like, those who live those healthy lifestyles, they're going to enjoy it. They're going to enjoy having social connection. I'm curious, as you built NeuroAge and you talked about how stress is bad for our brains, so both you and I are just maybe putting ourselves at higher risk. As you built NeuroAge, what is kind of the long-term goal? Obviously, you've got this great platform for testing, for understanding the risk of your brain, but what is the long-term goal for you guys?
Yeah, we were creating therapies to reverse brain aging. So eventually, what we want is to have a statin for Alzheimer's. So something that's safe, a pill that people can take that is going to prevent Alzheimer's in the future. And for that, I think we need personalized medicine, we need better data. So by taking the NeuroAge test, people are contributing in a privacy protected way to research that allows us to have better medicines in the future.
Yeah, I think that's awesome. I love what you guys are working on. I'm really excited that we're partnering with each other and that our members are going to be able to use your tests and really be able to use what they can afford. And you've been awesome to work with. And thank you so much for having this discussion with us today. This was great.
Yeah, thank you so much, Jared. I love Bionic, so I'm really happy that we have this partnership. This was a lot of fun.
Awesome. Thank you for listening to another episode of Medicine 3.0. We make this podcast so that you know where to turn for good information on keeping you and your loved ones healthy. Feel free to check us out at bionichealth.com, where we can help you live a longer, happier life.